Thank you, Dr. Holmes, for the introduction. And to begin with my thesis defense, I would like to express my gratitude to my thesis committee. My major advisor, Dr. Gerald Holmes, thesis committee, Dr. Shashika, Hewavitharana, and Dr. Dan Lagarde. I appreciate their support and guidance throughout the research project and my education. My topic today is strawberry disease management improvement for Macrophomina root rot and Botrytis fruit rot. I will.
Can you admit that might keep happening? Sorry.
All right.
Can anyone else get on the Zoom and moderate that order? So she can maybe on the laptop. If you could get on the Zoom, you could let people in, and she could not worry about it. Well, I don't know. Maybe you can't.
Yeah.
Unless you make her the host.
Yeah, that's okay. I can monitor.
He does everything.
As well. So first of all, I want to talk about how important the strawberry industry is here in California. So actually, California produces over 88% of the production in the United States. It is the top 5 commodity in California with more than 2.2 billion value. The major production area here is in the coastal area. From the north, we have Salinas, Watsonville. Coming down, we have Santa Maria and Oxnard. However, the production of strawberry is challenged by a lot of plant diseases. And that might reduce the yield and reduce the quality. A plant disease might be caused by microorganisms such as fungi, bacteria, nematodes, virus, and so on. And that could occur on different parts of a plant. Here I'm showing strawberry disease on the leaf part, on the fruit parts, and on the below ground, such as the crown and the root area. There are 2 economically important diseases I'm working on. One is Botrytis fruit rot, and the other is Macrophomina root rot. So I'm gonna begin with the first part of my study on the Botrytis fruit rot survey. And I believe this is a disease you guys have seen before. That is exactly the molded fruits that you see in the fridge if you leave your strawberry and raspberry in the fridge for a long time. So here I started with the Botrytis fruit rot disease cycle in strawberry. I begin with the surviving structure. So Botrytis pathogen may survive on the mycelium in the plant debris here, or the mycelium may aggregate together into a hard, rigid structure called sclerotia. This sclerotia structure allows the pathogen to survive over winter under harsh environment. Continue when the environment is allowed enough moisture for the pathogen to grow out. The sclerotia germinates and produce a large amount of spores. These spores could be dispersed by wind, splashing of water, or other human activity. And strawberry flowers is very susceptible to Botrytis fruit rot. So the pathogen will go onto those floral parts and infect, and the pathogen might stay dormant. Once the environment is good, you start seeing the lesion coming down from the calyx position, where the calyx could keep a very good moisture, and that is a good environment for the disease. And contacting infection is also occurring. Since Botrytis has a very wide growing temperature, they will continue growing under the cold storage condition. And that is the fruit you see in the store and you don't want to buy it. I included in this slide is a disease triangle. A disease is occurring with the presence of a susceptible host. Here we have the strawberry and the pathogen, Botrytis cinerea, and also an optimum condition. So the optimum condition for Botrytis is with the temperature 62 to 77 degrees Fahrenheit. and also a prolonged free moisture, which is more than 13 hours. That could be from the rainfall, the fog, the dew, or the irrigation. Fungicide is one of the most effective and common methods to protect our fruit. However, as I mentioned before, botrytis strawberry is continue having flower over time, and it is very susceptible to the disease. So fungicide application is demanding here. A survey actually find out over the season, we have 15 applications for botrytis fruit rot, and the application interval is 12 days. Frequent application of fungicide will exert strong selection for those resistant pathogen. So fungicide resistance will develop, and the fungicide will no longer be effective anymore. Also, application of fungicide is also costly. Based on a conversation with our grower, he tells us one spray is about $80 to $100 for an acre. Currently, there is a tool to guide the grower to spray fungicide when it is only the weather conducive environment. It is a strawberry advisory system made by University of Florida. The system had been validated in lots of places, such as Florida, South Carolina, Mid-Atlantic area. The research shows that if the grower follow the advisory system, they can reduce fungicide use for more than 18 to 50%, depends on the condition, and also not promising the yield. This is very promising here to know that. And we can see that on the map. These are the area they have the system active. Each circle indicates a weather station that is having the system working on and providing real-time risk regarding the disease. A is for anthracnose and B is for botrytis, and the color indicates the risk. So here, if it's showing green, that means the risk is low. If it's showing yellow, that means the risk is moderate, and if it's red, showing the risk is high. So for botrytis, as mentioned before, leaf wetness duration and temperature are two important components for the disease to occur. So when it's moderate risk, meaning the leaf wetness is about 14 to 18 hours leaf wetness duration, and high risk is more than 18 hours. So this system is continue providing the history of the risk factor over time and also providing some forecast information for the grower to make fungicide application decision. I am having somebody to join. Okay. So as we all know that California is with Mediterranean climates with very dry summer. And knowing that botrytis fruit rot is a disease that favored by free moisture. So we are wondering whether the frequent fungicide application is necessary here in California. To answer that question, we conduct a survey in growers' field. We want to compare the botrytis fruit rot incidence in growers' field with fungicide application, which is the grower's standard practices, and also what if he doesn't apply any fungicide at all. And we also want to evaluate those site-specific weather data using the model and to see the predicted spray and the actual spray number and what are the differences. Here are our experimental sites in Santa Maria area. We have this project in 2 years, 2021 and 2022. And the location here for 2021 is in blue pin here. We have field 1, field 11, field 31, and field 30 here. And for 2022, it's the red pin here. We have field 1, field 6, field 12, field 30, and field 31 here. And notice that the majority of this field, they are conventional practices. Only field 30 is an organic farm. And close to each location, we also install a weather station. The yellow pin is showing the location of the weather station. And since field 30 and field 31, they are very close to each other, they are sharing one weather station. all these fields, they are planted the Monterey cultivar, and the locations seen here, they are all over Santa Maria. Field 1 is the one very close to the ocean. It's only 2 miles away, and field 31 is about 20 miles away from the ocean. In each field, the grower saved 7 beds, which is about 0.2 acre, that they don't spray any fungicide that is targeting for Botrytis fruit rot for strawberry. And the rest of the field is their standard practices. We have 2 kinds of Botrytis fruit rot assessment, the in-field and the post-harvest. For the in-field one, we go into the field every week before the grower actually harvests to take samples. We harvest 50 fruits at 4 spots for each 2 treatments. So this figure actually indicates the area where we harvest those fruits. For the 7 beds of no fungicide treatment, we didn't harvest them at the edge of the field, and we did not harvest them right next by the fungicide bed to avoid any chance of fungicide drifting. And the black area is where we make the sampling. Right after sampling, we remove the calyx to evaluate the fruit to see if there is any incidence of Botrytis fruit rot incident. And here this picture showing the initial stage of Botrytis fruit rot just developed underneath the calyx. And then we calculate the Botrytis fruit rot incidence. The next assessment is post-harvest. This assessment was made by going to the field when the grower actually having the commercial picker pick, and we get 2 boxes of those marketable fruit from them, and transfer them to cow poly and store them for 7 days under cold storage, and conduct the Botrytis fruit rot assessment and calculate the Botrytis fruit rot incidence. Okay, here is the result. Due to the time limitation, I will only present field 1 and field 31, the raw data for 2021. And field 1 is represent the coastal field, and field 31 is represent the inland field. Somebody's joining. Okay, field 1, in-field Botrytis fruit rot incidence. Here we see the sampling time from May until October. And the orange bar showing the incidence of no fungicide application, and the blue bar showing the incidence of fungicide application. We can see that we don't really see a pattern, like which bar, the orange bar have high incidence or the blue bar have high incidence. It's altering, it's changing all the time, and the incidence is below 10%. I need to mute somebody on my Zoom, so that's why. And also for the post-harvest Botrytis fruit rot incidence, we see here I mark 2%, which the USDA #1 standard, and no fungicide in the orange bar, and fungicide in the blue bars. Here we see they are lower than the in-field incidence data, and a lot of them are below 2%. They match the USDA #1 standard, and some of them goes above that, but it's around 5% if they go above 2%. And for the in-field, field 35, the in-field Botrytis fruit rot incidence showing here, you guys see a lot of 0 here. It's a drier field, the incidence is low in the field, and also it's low in the post-harvest as well. So overall, the summary of the results for 2021, this graph shows the average Botrytis fruit rot incidence for the in-field and post-harvest incidents. We can comparing the fungicide incident in blue and no fungicide incident in red. For each individual field, we don't see a differences between fungicide or no fungicide spray. And if we combine all the conventional field, those data, we don't see any differences between fungicide and no fungicide, no matter it is in field or post-harvest as well. For the in-field data, we have the incidence of about 2.5%, and for the post-harvest, it is about 1.5 to 2%. We also see a pattern of the incidence reducing from the coastal area to the inland area. Here, that is more obvious for the inland Botrytis fura incident, and for the post-harvest, we can see a little bit of pattern as well. And seeing that the risk factor calculating by the strawberry advisory system, we also see the pattern of field 1 having the highest incident and reducing to the coastal area, with field 11 having a very low incident, which is very interesting. And this risk factor is corresponding to the daily leaf wetness duration showing here. So in general, field 11, a coastal area, we have about 15 hours of leaf wetness per day that indicate the dew event and some morning shower, and going to the inland is a lot drier. For 2022, here is a summary again showing the average botrytis incident in field and post-harvest. Notice that we have field 1, field 6, field 12 in the coastal area, and field 31 and 30 in the inland farm. But very interesting, we don't see a pattern that's a disease decreasing from the coastal to the inland. Here for the infield data, we see that field 31, inland farm, is having the same disease level as field 1, which is a coastal, very wet area. And field 6, located in the coastal area, is showing very low disease level. Coming here, we see the weather condition, the risk factor, is actually reducing when we go from the inland. We go from the coastal to the inland. So what is making this changing? One hypothesis is the plant canopy size. Plant canopy size might affect microclimates such as the airflow, the humidity where the fruits are located at. So we go into those fields to measure the plant diameter and plant height. And we actually find out field 31 and field 12, they have significantly higher plant heights and plant canopy. And field 6 is having smaller plant size. So that might be the reason why we have higher Botrytis fura incident in field 31, although it is located in a dry area. Regarding the first objectives, are there differences between fungicide and no fungicide application? There were no differences for the in-field Botrytis incident if we compare that for individual fields. But we see the post-harvest Botrytis fura incident having some differences. For field 1 and field 12, we see that the no fungicide is having higher incidence of Botrytis fura. And that is showing for when all the conventional data are combining. The post-harvest Botrytis incident with no fungicide showing high incidence. However, it's still very low. We see that it's about 3 to 4% for the infield and about 0.5 to 1.5% for the post-harvest. Okay, moving on, our next objective. We would like to apply California weather data to the disease model made by Strawberry Advisory System. So showing the raw data here, field 1 in 2021, we see that over time, the incidence in the bars and the risk factor here showing in the dotted line. We can see that applying them to the risk factor, green for the low risk, yellow for the moderate risk, and red for the high risk. And we can see that sometimes it goes to high risk. And adding on the growers' fungicide application practices. So here we see that the grower is spraying fungicide in a pattern for about 2 weeks interval, and Cap 10 is the most common fungicide they are using. And comparing the differences, their actual spray is 7, and our predict spray, when it's go above 0.5 in the risk factor, is 7. For the inland dry field 31, we also see the dotted line for the risk factor and applying that to the risk low, moderate, and high. We see that it never go above to moderate or high risk in this field. However, growers still apply some fungicide in this field. For the fresh fruit market, they have 3 fungicide application, and the predicted spray is only 0. So summarizing here, what is the potential saving if the grower follow the strawberry advisory system? I calculate that for 2021 by making the differences between the actual spray and the predicted spray. And we found that if the grower followed the strawberry advisory system, they could have about 50% of reduction in 2021, which is about 71,000 for those 4 fields. And for our current available data for 2022, it's about 14% of reduction. And regarding how well the risk factor is predicting is associated with actual Botrytis fruit rot incident here in California, we built a regression model to have this result. In 2021, it was a great fit, and in 2022, it wasn't a great fit. It might be because of the plant canopy issue affecting the result. Here, concluding the results over the 2 years, our survey in Santa Maria, we found that low disease incidence is occurring no matter it is with fungicide or without fungicide. In 2021, we have an infield incidence about 2.5%, and post-harvest is about 1.8 to 2%. And in 2022, it's about 3 to 3.7% for infield and 0.6 to 1.5% for post-harvest. This is very low, and seeing that it happened for the fungicide and no fungicide application. So this is telling us there might be a lot of chance for us to reduce fungicide use. But reducing fungicide use may increase the farm profits and also extend the effectiveness of fungicide. And in both years, we found out plant canopy may alter the disease incidence. So cultural practices to alter the plant canopy may be applied to control the disease. So the grower can manipulate the chilling hours of the transplant or changing the nitrogen fertilizer schedule to avoid those vigorous vegetative growth. And of course, the impact of using strawberry advisory system here is need to be further investigated, and this research should be further conducted in different years with different weather conditions. Moving on to the next disease, a soil-borne disease called Macrophomina root rot. Before I start, I would like to know how many of you have planted a strawberry plant before? Oh, the majority of you. Okay, that's very nice. So we all know that there's not only the above ground diseases, also the below ground diseases when we plant the strawberry into the soil. So Macrophomina root rot is one of them. So here, Macrophomina root rot is caused by the soil-borne fungus Macrophomina fasciolina. As I mentioned before, not like Botrytis fruit rot, this is a pathogen living in the soil and attacking the crown and root area. Strawberry plants infected with Macrophomina root rot show symptoms of stunted, wilting, the above ground get necrotic, and eventually you see the plant collapse and producing no yield. And if you cut the crown open, you will also see the dark discoloration showing the crown is being affected. It is a disease we found a lot in California strawberry industry. For our disease diagnosis service we provide to the grower, we found it in about 22 to 26% of our samples. And this disease is favored by high temperature and low soil moisture. Since Macrophomina root rot is a relatively new disease in strawberry, but it's a more well-known disease in chickpea. Here I am showing the disease cycle of Macrophomina root rot in chickpea, which is made by Ray et al. In 2022. And I will accompany by some strawberry plant pictures. So here, the primary inoculum is the microsclerotia. This is a dark, rigid structure that's formed by the aggregating of mycelium, and this allows the disease to survive in plant debris and soil for a long time. And moving forward, after we plant strawberry into the soil, the latent infection actually occurs right away when the root tissue encounters the microsclerotia. So the root exudate will stimulate the growth and the germinate of the microsclerotia. So the microsclerotia can germinate and grow, penetrate into the root tissue. And further infection, we see they are growing and colonize the root tissue more. And you start seeing the partial corrosis, necrosis on the above ground tissue that happen when it's during drought condition especially. And further, you see the disease development more, and the disease is producing some daughter microsclerotia in the root system, and eventually the root rots and the plant collapse. Soil fumigation is one of the most common and effective methods to manage soil-borne disease. However, due to current policy restrict some effective soil fumigant, we are looking for some alternative management method, such as host plant resistance. There are some strawberry breeding program breeding strawberry cultivar against Macrophomina root rot. And here I'm showing the cultivars from California, University of California here, and their resistant category for Macrophomina root rot. Resistant is showing in green here, and susceptible is showing in red here. And you can see that the majority of our cultivar, they are susceptible to the disease, and there's no complete resistance to the disease. So there is a high need for us to build a strawberry that is resistant for Macrophomina root rot. Here at the Cal Poly Strawberry Center, we have been conducted a cultivar evaluation here for 6 seasons so far, starting 2017 until this year. We continue obtaining new genotype from different breeding programs and evaluate their susceptibility to the Macrophomina root rot. I have 3 objectives here, including, and I will go over each of that. So the first one is we would like to evaluate the susceptibility of strawberry cultivar and elite breeding lines, and that is for the latest 2 years. First of all, the experimental design. We conducted this field in our field 35. We have 51 genotypes in 2021 and another 51 genotypes in 2022. Notice that there might be some different genotypes since we obtain new genotypes from the breeding programs every year. And these are the 4 and 5 to 5 breeding program that was involved in these 2 years. And the area view is showing the design of our experiment. We have 4 replicates that we actually artificially inoculate every single plant here. And we also have an additional reference here in the fumigated area that we plan 1 replicate, and we want to see how they perform. The timeline of the event is similar for each season, so I will take 2021 season for example. The bare root transplant were planted on November 2nd, 2020. And 2 weeks later, we introduced artificial Macrophomina inoculum to the roots and crown area for every single plant. We want to make sure the disease level is similar for every single plant and every genotypes. And starting on December, we conduct mortality assessment for 2 weeks. We consider 100% necrotic for the mortality. Because the plants may die for other reasons, such as another biotic factor or another plant disease, we conduct molecular technique and plating technique to confirm the causing agent is Macrophomina root rot. If it is not, we will exclude that for our data. And to further induce the disease level, we introduce drought stress starting the June 1st. Okay, here is the final disease rating at the end of 2021 season. We see some elite breeding lines showing very vigorous by the end of the season. And Monterey, which is the most commonly grown variety, showing some stunted, corrosis, and Ruby June is a very susceptible cultivar. Over those 51 genotypes, we see a wide range of mortality from 0% to 67%, with an average of 15%. And I pointed out those cultivar from UC and their resistant category. Green is resistant, red is susceptible. They are majority match our resistant ranking. Just in our study, Monterey is showing more resistant than UC D Victor. Another year, the final disease rating on August 2022, we see some cultivars showing more vigorous, more resistant looking here, and some cultivars showing susceptible looking here. Also, a wide range of susceptibility was finding. The mortality rates go from 3% to 45% with an average of 18%. Again, I pointed out some UC cultivar. They are matched the order to our study as well. Just Monterey is showing more resistant than UCD Victor as well. So moving on to our next objective, we want to investigate some temperature effect on disease severity. So the reason why we want to do that is actually because we see disease severity varied among years. As I mentioned before, this is a study has been conducted for 6 years. Every year we see the final disease mortality varied. Here, the 4 common cultivar here and their plant mortality shown here, and we see their disease mortality varied. And we wonder what is causing this happen? And knowing that the disease is favored by heat, we want to know more about the temperature effect. So we evaluate the soil temperature and the air temperature considering the monthly factor, seasonal factor, and cumulative temperature effect on the disease severity. Among all temperature variables we explore, we actually find out the November soil temperature, which is a planting month soil temperature, is highly associated with final disease mortality. Here we see the soil temperatures goes from 59 to 62 Fahrenheit, and the final disease mortality on the y-axis, and each dot represents each year's data for those 8 cultivars. We see the relation actually depends on cultivar. So the 3 cultivars, they are having very good fits, R-square value higher than 0.9. And the 3 cultivar here, they are poor fit, R-square value lower than 0.5. But in general, this relationship is having an R-square value of 0.79, which is saying that it's explaining 79% of the variation. So considering this is a field research, this is very high number, saying that how important soil temperature is. Besides that, we also find out the mid-season air temperature of April is also highly associated with the final disease incidence. Looking here, the air temperature of 55 to 63 degrees Fahrenheit and the final disease mortality here for those 8 cultivars. We find out some cultivar, they are a great fit in this relationship, and the rest of them are just moderate risk. But overall, this relationship is also giving us R-square value 0.79, explaining 79% of the variation. The next objective, the final one of this project. Since we have been conducting this research for 6 years, I want to summarize some susceptibility, resistance of some cultivars. And for 4 seasons of 2017 to 2020, I can find out the most amount of genotypes, since we always obtain new genotype for every year. So for these 3 years, I can conclude 14 cultivars here. I categorize them based on the average final mortality over the 4 seasons in 4 resistant category. Resistant here showing is when the final mortality is below 22%, and moderate resistant is when it's 22% to 45%, and moderate susceptible is when it's 45 to 68%, and susceptible when it's below 90%. This value are from divided 4 by the highest number I have, so they are equal for each range. And fitting them to our 14 cultivars, I fit them by the average mortality over the 4 years, and it is showing in the dot here. So among those 14 strawberry cultivars, only 1 cultivar, Osceola, is being categorized in the resistant category. And there are 7 cultivars in moderate resistant, and 3 in moderate susceptible, and 3 in susceptible. Notice that Monterey and Fronteras, which are the most commonly grown 2 strawberry cultivars, they are in moderate susceptible category in this study. Finally, I would like to conclude some findings from this research. We see a wide range of susceptibility that is existing in all breeding programs. Some of them have resistance, some of them have susceptible, but that exists in all the breeding programs. They might be very good breeding materials for them. And I conclude some resistant information. Some of them are in resistant or moderate resistant category, but notice that there's no complete resistance exists in this trial. Since we continue getting new genotypes from the breeders, the breeder is combining the soil-borne disease resistant data with other horticultural traits. Our resistance screening should be ongoing for those genotypes for more years. Another aspect is our understanding more about the biology of the disease. The disease is affected by soil temperature of the planting months and air temperature at the mid-season. So in a year with warmer temperature, we might see the disease become more severe, and in a cooler year, we might see the disease is less severe. Finally, these projects are funded by the California Strawberry Commission and USDA NIFA, and also the strawberry growers Brian Grasser and Greg Friends. I appreciate the growers allow us to go into their field to sampling and to apply the no fungicide treatment in their field. And again, this is a teamwork effort. Many thanks to the personnel listing here. They are very helpful to this project. The staffs from the Strawberry Commission, especially the Santa Maria Field Research Team, and also Cal Poly Strawberry Center staffs, student, undergrad student, grad student, and 4 more postdoc. Thank you all for your effort and your hard work to make this project possible. Thank you all for your attention, and I am ready to take any questions you might have.
Thank you, YU-CHEN, for an interesting research seminar. And we have a few minutes to entertain questions from the audience before we have a defense with the committee. And it looks like there's some questions already. Jim?
Yeah, really nice job. I have a question. If the soil temperature is so important for Macrophomina when you plant, would a recommendation be if the soil temperature at your planting date is too high to even delay a week or two, or would that too negatively impact the growth of the strawberries during the productive season?
This is a very good question regarding the compromising of the growth and regarding the disease pressure. But I think for this project giving us, if the grower is having Macrophomina history in their field, they may want to watch the soil temperature when they are actually planting. So I was discussing with some other researchers. They are talking about maybe putting out irrigation before they plant. That can reduce the high temperature before they plant, or they can maybe change the mulch color from white or black. That might have another impact. But that may be only for the grower who has Macrophomina issue, because the soil temperature could also promote the plant growth as well. So that is a very interesting study we may continue studying. But thank you for your question. I wish I answered that. Okay, thank you.
So for all the fields that you sampled, were the growers planting the same variety?
Yes, they are all Monterey variety.
Okay, and then for the fungicide treated, like the field part that you treated, and then your untreated one, did you run, for the incidence, did you run any correlation tests and see the incidence, whether they correlate?
Between fungicide?
Fungicide treated and non-treated. So for example, sometimes you see a higher incidence in treated part, sometimes you see a higher incidence in the non-treated part. Did you run a correlation test and see, despite of that, whether they are correlated, the incidence?
So are you saying the timing regarding each time they treat fungicide? So that will be very interesting if we look at this graph regarding the fungicide and the fungicide timing and the disease incidence. I think this is what you are referring. Like if they treat fungicide, if the disease incidence actually reducing or not, right? Like at the timing. But actually, I don't see that in our data.
Let me see if I could clarify. Are you saying, so in other words, if you have high disease and no fungicide,
right,
would you also have a somewhat
right,
high, in other words, these things are moving together even
right,
if they're,
right,
so
right,
when you have high levels in the no fungicide, you'd also
right,
have high levels in the fungicide.
right, right.
And vice versa, if it was low, it would also be low. And
oh, thank you.
So for each period, then you'd have to correlate that over time. I don't think that's something that you did.
Yeah, that is not something I did, and that is very good comment. And that, yeah.
Because that way you can see how the weather pattern influenced whether it's treated or not.
Yes, yes, thank you for that comment. And we did not do that, and that would be a very good practice to know if they have correlation.
I think it could be a simple Pearson correlation. Yeah. and then I do have another question. So the forecasting, what is that? Strawberry status? Can't remember the abbreviation. But is that based on previous weather or based on forecasted weather?
Both based on the real-time weather we are collecting. And regarding your first question, I did something kind of similar, but I was making correlation between the risk factor and my incidents. So risk factor indicate the weather condition of it's wet or not, the temperature is fit or not, and for 2021 they have good correlation, but not for 2022. And that reason might be because of the canopy effect I mentioned.
You probably can do the same for
Each year, yes.
and non-treated.
Yes, thank you for that comment.
Good.
Yeah, so great presentation and some nice body of research that you've produced over the last couple of years. So I've got a question on your Macrophomina study, and this is more of a kind of a theoretical question, but do you have any ideas on what might be some of the sources of resistance or the mechanisms of resistance in the cultivars that you found to be less susceptible than those that were more?
So until like 2021, there's just a paper published, some QTL, and highly associated with the resistance of Macrophomina root rot for strawberry. And that was a paper published in 2021 by Nelson. So there's some new finding regarding the resistance.
So is it something intrinsic to the plant, or is it related to root exudates, or?
It's quantitative.
Like, not the level of the gene necessarily, but the.
So that one I mentioned is regarding the resistance, and others such as like the structure or other way the plant tolerance the disease. I have not seen any other research. Thank you for the comment.
Let's go Kyle and then Jen.
On the Botrytis project, you showed the cost savings of grower could have from year 1 to year 2. I think year 1 was 50% savings and year 2 was like 14. Do you know why there was such a difference between the 2 years?
So the second years, we actually have the fungicide data only until April to May. So we don't have the whole data set since the data updated needs some time to the Ag Commissioner office and to download. So we don't have the full data set. So that is just partially.
So potentially it could be a lot higher savings then for the full season then?
I cannot make the comments, but hopefully.
It's possible though, right?
Yeah.
Because we just don't have the data yet. Jack?
Regarding the BFR incidence trial, you mentioned that all of the cultivars that you were examining were Monterey, but then you also stated that there were canopy differences. So were there different planting times? Were they progressing at different rates? And do you think that that could have played a role in your incidence levels?
Yeah, thank you for the comments. So they are all fall planting for all those fields. And as I mentioned before, cultural management may be one very important way to manage the plant canopy. So by applying nitrogen may promote a lot of vegetative growth, or the chilling hours. So the chilling hours for the transplant, there is a optimum hour of 150 to 400. So if that goes above that range, the plants will go vigorously, vegetatively. So there are some cultural management regarding why there are differences between each field, and although they are the same cultivar.
For your MAC trial for the Petri dish study, when determining cause of death, whether it's Macrophomina or something else, how did you decide whether to dictated being macrophomina or not, if you had multiple pathogens that came up either in the Petri dish or in a mud.
Thank you for that question. So for my case, I don't find several pathogens at the same time. So usually like during the winter time, I find a lot of Pythian, Phytophthora, and by the season, during the summertime, I only find Macrophomina. So I wasn't finding several diseases at the same time in our field, so which is easier for me.
So you said that for the Botrytis project, for the inland fields, I think this was 2021, you found 0 predicted sprays with the model versus 3 actual sprays that happened, versus the more coastal, you saw 7 predicted and 7 that actually happened. So did you see that for the inland fields where there were more actual sprays than predicted, did you see there that the non-sprayed fared better compared to the sprayed than in the coastal area? Does that make sense? Sorry, that's a long question.
Yes.
Does that make sense?
That is a very, very good question. I have not think of that way, but the differences is very small for the inland, I believe. So I can look up more, but that is a very good question.
YU-CHEN, are there any known genetic resistances to Botrytis fruit rot? And if not, are you guys planning on screening for it?
Thank you for that question. So actually, the breeding program mostly focus on horticulture traits, such as like day neutral, short day variety, or how sweet the fruits are. So not until recently, the soil-borne disease become their focus. And regarding the question whether Botrytis is their breeding program focus, I don't think so from my literature review. But there are some researchers comparing the fruit, different variety of fruit, and how that resistant to Botrytis. I have seen that kind of research, and some of them have field study as well, but I don't think that is a major goal for those breeding programs so far.
But they have found some differences.
Yes,
anyway.
yes. Mm-hmm.
Just one last question for me. Just maybe the same question as Dr. Ding asked, but I wanted to clarify that in the Botrytis project where you're presenting the Botrytis fruit rot post-harvest data on a weekly basis, you didn't run any statistical separation to see if there was a difference between fungicide and non-fungicide?
Yes, so for each week, I did not do that. I did that for the whole season. So my analysis is based on all the sampling time together, represented each samples. I did not do that individually. And again, I don't really have a true replicate in this project because this is in a grower's field. It's really hard to create those kind of true replicate. So I did not do that comparison. It's because that restriction as well.
All right, any other questions? Before we
What about questions?
Yes.
do you have questions on Zoom? I wanted to let everybody know that YU-CHEN actually has a job waiting for her, and all of this work that she's done and her previous jobs and her other master's degree have prepared her very nicely for a position in cooperative extension. She's starting in October as the new farm advisor in plant pathology in Santa Cruz, Monterey, and San Benito counties. So we'll see more of YU-CHEN. She'll be working on all the crops in the Salinas Valley and that area of the state, so including strawberries.
Mm-hmm.
Are there questions on the Zoom for you? You want to entertain, or?
Yes, I don't think there are any questions, and if anybody can hear me and you want to ask a question on the Zoom. Yeah, I don't think there's any question.
All right. Well, let's thank YU-CHEN.
Thank you.